Welcome back. In the past several weeks, we have looked at various aspects of drug discovery. So, we have looked at how to optimize the target drug interaction by doing structure activity relationships. We have looked at how to develop assays for testing the efficacy of a molecule. We have looked at how to generate libraries of molecule. So, for example, in the just recently we were looking at combinatorial chemistry, which produces mixtures of of compounds. We have also looked at parallel synthesis, which gives us pure material, but a fewer number of compounds. And of course, we have looked at natural products as sources of drugs. And so, all of these gives us a very strong foundation about how to carry out drug discovery. So, in the next 2 or 3 weeks, what we will focus on is to divide the drug discovery in terms of the diseases. So, the major diseases that we are going to encounter in terms of drug discovery are basically bacterial infections. So, as we are all familiar, bacterial infections are something that affects nearly everyone, and so there are a lot of antibiotics that are available for treating such infections. And so, the first topic that we will take up is antibacterial agents, and then we will look at antiviral agents. And as we perhaps are familiar with, viruses are again very common, and there are actually very few drugs available for viruses, but we will look into these aspects of how antiviral drugs have been discovered, which will be followed by another major disease which affects nearly everyone, which is cancer. So, cancer is a situation where there is uncontrolled proliferation of cells, and therefore, it presents a major challenge because there is a lot of similarity between cancers and human cells. And so, we will look at various aspects of anti-cancer drugs. And lastly, we will look at all the cholinergics and adrenergics, which are important in various diseases, including cardiovascular diseases and all that. And finally, we will look at quantitative structure activity relationships, which is basically one of the attempts to model a structure activity relationship and to construct equations which show the various aspects of properties of a drug versus its activity. Now, what we will do is we will start with antibacterial agents. So, we will present the major classes of antibiotics and how they were discovered, what is the mechanism of action. So, this part of the course will be handled by Anand.
As you are now familiar with various concepts in the medicinal chemistry course, let us take it to the next level, where we are going to deal with one of the well prescribed known class of drugs called as antibiotics that target the bacterium. We live in a world filled with bacteria. These are so tiny organisms that we cannot see them through our naked eyes, but they are there and they are literally everywhere. Actually, bacteria aren't all bad. There are many good bacteria which are responsible for digesting the food in your gut. There are bacteria which are helpful in making medicines. There are bacteria which are essential for making dairy products, which some of you may like to eat. Believe it or not, there are billions of good bacterial cells in your body without which you wouldn't be able to survive today. But there are a handful of bad bacteria as well, and they are called as pathogenic bacteria, and they are responsible for many pathogenic infections. They are more like they want to multiply by number and use your body to spread infections into an environment. So, that is basically what they do. So, they are responsible for all these infections. Fortunately, these pathogenic bacteria can be knocked out from your body by the so called fantastic drugs known as antibiotics. The word antibiotic literally means against life. So, the goal of the antibiotic is either to kill the bacterium or it is going to inhibit the growth of bacterium. So, these antibiotics can work in either way. So, they might be called as bacteriostatic antibiotic or bactericidal antibiotic. Here, the bactericidal antibiotics are those which are going to kill the bacteria. If you see, the suffix cidal means kill. There are some antibiotics which are bactericidal antibiotics like aminoglycosides, cephalosporins and fluoroquinolones. But there is another group of antibiotics called as bacteriostatic antibiotics. Here, the suffix static means staying stable. So, in these bacteriostatic antibiotics, bacteria don't die, but they can't grow or replicate either. For example, tetracyclines, chloramphenicol, macrolides and lincosamides, all these are bacteriostatic antibiotics. There is another class of antibiotic which can be differentiated on the terms of spectrum of activity. For example, They are antibiotics which are highly effective against gram positive organisms. Here in this cartoon, this is a gram positive bacteria, and this is an example of gram negative bacteria. Do not bother about the terms gram positive and gram negative for the time being. We will discuss in detail in the next couple of minutes, but for the time being, please do remember there are some antibiotics which are highly effective against these gram positive organisms because they have very simpler cell wall structure. So, these antibiotics can easily get inside the gram positive bacteria. For example, macrolides is a narrow spectrum antibiotic which is only effective against gram positive organisms, but there are some antibiotics which can kill both gram positive as well as gram negative organisms. For example, aminoglycoside. Other than these classification, there is another major classification of antibiotic based on the route of administration. For example, there are some antibiotics which cannot be taken via oral route. The reason is they are susceptible to acid that is present in the stomach, and therefore these antibiotics will undergo degradation. So, in such cases, these antibiotics must be entered through intravenous route of administration, where you can inject the given antibiotic directly inside the vein so that it can reach into the bloodstream. Penicillin V is one of the example of orally active antibiotic. That means you can give in the form of a tablet and it can be taken via oral route. Penicillin G must be given via intravenous route of administration because it is highly susceptible to stomach acids. And now you are familiar with the classification of antibiotic based on bacteriostatic or bactericidal antibiotic. Again, these antibiotics can have either narrow spectrum of activity, that is they are highly effective only against gram positive organisms, and other one is broad spectrum antibiotics where they are effective against both gram positive and gram negative organisms. But basically each antibiotic have their unique mode of action. Ultimately the job of the antibiotic is to kill the bacteria, or it is going to inhibit the growth of bacteria, and this can be done via this different mode of action. Some antibiotics are going to inhibit the cell wall biosynthesis, some are going to target nucleic acid biosynthesis, some are going to target protein biosynthesis, and some are going to affect the metabolic pathways of the bacteria. In this lecture, we will be mostly focused on how these antibiotics are going to kill the bacterium or inhibit the growth of bacterium by these unique mode of actions. This is again broad classification of all the antibiotics according to their mode of action. As I already mentioned you, there are some antibiotics which target the cell wall biosynthesis, for example, beta-lactam antibiotics and glycopeptide antibiotics. Polypeptide antibiotics is an example that disrupts the cytoplasmic membrane. Quinolones and fluoroquinolones are another class of antibiotics that target the nucleic acid biosynthesis of the bacteria. Majority of the antibiotics are going to target protein biosynthesis such as tetracycline, macrolides, aminoglycosides, chloramphenicol, etc. Very few antibiotics target the metabolic pathways such as folate biosynthesis. So, this is the graphical abstract which tells how each antibiotic have their unique mode of action, and ultimately they are going to either kill the bacteria or inhibit the growth of bacteria. Now, let us continue with the topic on the antibiotic that target the cell wall biosynthesis. So, cell walls are very good targets for antibiotics because bacteria need them for their survival and our cells do not even have them. So, let us briefly review what these bacterial cell walls are. So, bacterial cell walls are very tough rigid structures that forms a protective outer layer around the bacterial cell, and it helps them to resist the effects of osmosis. Remember, the term osmosis is the tendency of water to flow across the membrane in such a way to balance out the number of solute molecules on either side. Without this bacterial cell wall, cell loses its rigidity and bacteria would burst and die due to osmotic stress. Different bacteria have different cell wall structures, just like the outside of buildings can have many different designs, but still fulfills the same purpose, that is keeping the outside separate from the inside and protecting the inhabitants from an environment that is sometimes hostile. Danish scientist Hans Christian Gram developed a strategy to differentiate the different types of bacteria based on their structural differences in the cell wall. In his test, the gram positive bacteria retains this crystal violet dye, and that is why they are going to appear in the microscope as violet color. One such example of gram positive bacteria is Staphylococcus aureus. In contrast, gram negative bacteria do not retain the crystal violet dye. Instead, they are stained by a counter stain called as safranin red, and that is the reason they appear as pink color in the microscope. E. coli is one such example of this gram negative bacterium. Now, what is the basic difference between this gram positive and gram negative bacterium? The only difference is on the basis of their cell wall structure. So, in case of gram positive bacteria, they have a very thick layer of peptidoglycan, which is again a molecule that is made up of sugars and polypeptides, which forms a very rigid mesh like structure that protects the bacterial cell. Gram negative bacteria also has a peptidoglycan layer, but this is very thinner layer that is sandwiched between two membranes. In addition, the outer membrane of the gram negative bacteria contains a component called as lipopolysaccharide, which further adds to the complexity of the gram negative bacterial cell wall. Due to the presence of this additional complex barrier, it is very challenging for the antibiotics to target the gram negative bacteria. Now, if you recall, I mentioned you about two terms of antibiotics, which is narrow spectrum antibiotic and broad spectrum antibiotic. I told you that narrow spectrum antibiotics are highly effective against gram positive organisms because the cell wall structure is very simpler. So, the antibiotic can easily get inside the gram positive bacterial cell. But in case of gram negative bacteria, because of the presence of this lipopolysaccharide, many antibiotics are unable to penetrate the gram negative bacteria, and therefore, the antibiotics which can target both gram positive and gram negative bacteria, they are called as broad spectrum antibiotics. As you are familiar that the cell walls are the perfect targets for an antibiotic. Now, what do these gram positive and gram negative bacterial cell walls have in common? It is peptidoglycan. So, let us look at the structure and biosynthesis of peptidoglycan. But before that, let me remind you that peptidoglycan is made up of repeating units of N-acetyl glucosamine, which is abbreviated as SNAG, and N-acetyl muramic acid, or NAM for short. Let us quickly look at the biosynthesis of peptidoglycan. Peptidoglycan is biosynthesized in three phases. The first phase is called as the cytoplasmic phase. So, here is the cytoplasmic phase. Then these components, once they are biosynthesized in the cytoplasm, they are translocated from the cytoplasmic side to the periplasmic side, and this is called as the membrane associated phase. Once these components are translocated to the periplasm, they are added to the precursor of the cell wall components, and that is how they are going to assemble this cell wall structure, and this is called as the extracytoplasmic phase. In case of this cytoplasmic phase, if you recall, I already mentioned you that peptidoglycan is made up of N-acetyl glucosamine and N-acetyl muramic acid. So, obviously, this UDP N-acetyl glucosamine is the precursor for peptidoglycan. So, now this UDP N-acetyl glucosamine is converted to UDP N-acetyl muramic acid by a presence of enzyme called as MUR-A enzyme. Once the UDP N-acetyl muramic acid is being formed, the pentapeptide chain is added to this, and this leads to the formation of UDP N-acetyl muramic acid pentapeptide, and this is the cartoon representation of that pentapeptide chain. This whole process is carried out by a series of enzymes which belongs to MUR family, from MUR-A to MUR-F. So, this is the cytoplasmic phase. Now, here if you see, it has a UDP component and it has a sugar moiety attached to it, because of which it is highly polar and hydrophilic. Now, the thing is, it has to be translocated from the cytoplasmic side to the periplasmic side, and which cannot be done without the help of any lipid. So, here in this case, the lipid which helps in translocating this component from the cytoplasmic side to the periplasmic side is C55 lipid phosphate. So, what actually happens is, the C55 lipid phosphate attacks on the pyrophosphate linkage of this UDP N-acetyl muramic acid pentapeptide in the presence of an enzyme called as MRAY, releasing this UMP and forming a new pyrophosphate bond between the C55 lipid phosphate and the UDP N-acetyl muramic acid pentapeptide. This is the first lipid intermediate that is being formed, and this is called as lipid 1. Then this is followed by addition of UDP N-acetyl glucosamine component to this lipid. This results in the formation of another lipid intermediate called as lipid 2. This process is carried out by an enzyme called as MUR-G. Now, once this lipid 2 is formed, it is translocated from the cytoplasmic side to the periplasmic side, and this is carried out by an enzyme called as translocase enzyme. No definitive evidence for this enzyme is yet available. Once it is been translocated to the periplasmic side, then it becomes a substrate for many membrane bound enzymes such as transglycosylase, transpeptidase, etc. So, here this transglycosylase cleaves this bond and releases this C55 lipid pyrophosphate and peptidoglycan component. If this cycle has to happen again, this C55 lipid pyrophosphate must be converted to C55 lipid phosphate. This pyrophosphate bond is cleaved in the presence of an enzyme called as pyrophosphatase, and then again the C55 lipid phosphate is recycled, so that this process can happen again and again. Here whatever the pentapeptide chains are there, so these pentapeptide chains are extensively cross linked by an enzyme called as transpeptidase enzyme. This process of extensive cross linking continues until it forms a very rigid mesh like structure of cell wall. This is the whole process how the peptidoglycan components are being biosynthesized and transported from the cytoplasmic side to the periplasmic side. Antibiotics inhibit the cell wall synthesis by targeting any of these steps. For example, fosfomycin is an antibiotic which inhibits this cytoplasmic phase by inhibiting the conversion of UDP N-acetyl glucosamine into UDP N-acetyl muramic acid. There are some other antibiotics like tunicamycin and muridomycin that are going to inhibit the translocase enzyme. Majority of the antibiotics that inhibit the cell wall synthesis either target the transglycosylase or transpeptidase enzyme. For example, beta-lactam antibiotics like penicillins and cephalosporins, they are going to target the transpeptidase enzyme. There is another class of antibiotic called as bezitracin, which targets this pyrophosphatase. As you are familiar with the biosynthetic pathway of the peptidoglycan component, let us look at the molecular structure of peptidoglycan. So, peptidoglycan is made from N-acetyl glucosamine and N-acetyl muramic acid, but it has a pentapeptide unit chain attached to it. And this pentapeptide contains amino acids such as L-alanine, D-glutamic acid, L-lysine, D-alanine and D-alanine. This is the composition of peptidoglycan component in a gram-positive bacteria, for example, Staphylococcus aureus. But in case of gram-negative bacteria, instead of this L-lysine, they will have meso diamino pimelic acid. This is the only difference between the peptidoglycan component of gram-positive bacteria with respect to gram-negative bacteria. It is interesting to note the presence of D amino acids in this chain. In human biochemistry, there are only L amino acids, but in bacteria, there is an enzyme called as racemase which can convert the L amino acids into D amino acids. In the final step of cell wall biosynthesis, the two peptide chains are cross linked, and this occurs due to the displacement of one of the D-alanine unit from the chain by the glycine unit of the other chain, and this whole process is called as transpeptidation reaction, and this is carried out by an enzyme called as transpeptidase enzyme. Due to presence of this transpeptidase enzyme, there is extensive cross linking between the peptide units of the peptidoglycan component, and that is how it forms a rigid mesh like structure around the bacterial cell, and it helps them to resist the effects of osmosis. Let us try to understand the mechanism of transpeptidase enzyme. Here the transpeptidase enzyme is an enzyme that is bound on the outer surface of the cell membrane, and it contains a serine residue in the active site of this enzyme. This is very much similar to the family of enzymes called as the serine proteases, so named because it contains a serine residue and they are involved in the hydrolysis of the peptide bond. Here in this case, the serine residue of the transpeptidase enzyme acts as a nucleophile, and it split the peptide bond between the two unusual D-alanine units on a peptide chain, and in this way the terminal D-alanine unit departs the active site, leaving the peptide chain bound to the active site. Next, the pentaglycyl moiety of another peptide chain enters the active site, and the terminal glycine forms a peptide bond, and displacing it from the serine, and this is how the two chains are cross linked with each other, and this process continues until it forms a very rigid mesh like structure on the bacterial cell. The transpeptidase enzyme is the specific target for beta-lactam antibiotics, which is one of the widely prescribed class of cell wall biosynthetic inhibitors. These beta-lactams include drugs such as penicillins and cephalosporins, and they are going to inhibit this transpeptidase enzyme. They are going to bind to this enzyme and irreversibly inhibit this enzyme, and that is how it is going to inhibit the peptidoglycan biosynthesis. Because of that reason, cell wall becomes very fragile, and eventually osmotic pressure builds up that the bacterium would burst and die. As a result, these beta-lactam antibiotics are bactericidal in nature, because they are going to directly kill the bacteria by inhibiting this peptidoglycan biosynthesis. Let us look what these beta-lactam antibiotics are. So, these are one of the most commonly prescribed cell wall biosynthetic inhibitors, which have a common structure as well as the mechanism of action, that is, they are going to inhibit the peptidoglycan biosynthesis. So, here is the structure of beta-lactam ring, which is a highly strained 4-member ring which consists of a cyclic amide. Now, these beta-lactams can be classified into 4 different group of antibiotics like penicillins, cephalosporins, carbapenems and monobactams based on the variation in their core ring structure. For example, in case of penicillins, they have a 4-member beta-lactam ring which is fused to 5-member sulfur containing dihydrothiazole ring, and they are called as penem. And if the 4-member beta-lactam ring is fused to 5-member saturated system, then they are called as penems. Remember, in both the cases the heteroatom present here is sulfur. Instead of sulfur, if it is an oxygen, then it is called as oxapenems. Likewise, instead of any heteroatom, if the carbon is present, then they are called as carbapenems. In case of cephalosporins, the 4-member beta-lactam ring is fused to a 6-member sulfur containing 2,3-dihydrothiazine ring, then they are called as cephems, and if it is fused to 6-member sulfur containing thiazine ring, they are called as cephems. Again, in both the cases, the sulfur is the heteroatom present in these class of antibiotics. If it is oxygen instead of sulfur, then they are called as oxacephems. There is another unique class of beta-lactam antibiotics which have only monocyclic ring and it is not fused to any other ring, and they are called as monobactams. So, this lecture will focus on detailed study of each of these class of beta-lactam antibiotics, and now let us start with penicillins. So, these penicillins are again beta-lactam antibiotics and they exhibit spectrum of activity from narrow spectrum to broad spectrum. They are bactericidal antibiotics because they directly kill the bacteria by inhibiting the peptidoglycan biosynthesis. It is derived from a fungus called as Acremonium chrysogenum, and these penicillins are used for variety of infections such as meningitis, which is associated with the inflammation of meninges that cover your brain and spinal cord. Likewise, penicillins are also useful in streptococcal and staphylococcus infections. Penicillins are also effective against a sexually transmitted disease called as syphilis. It is also effective against a bacterium called as Clostridium tetani, which is responsible for an infection called as tetanus, which is associated with severe muscle spasms. This is the basic code structure of penicillin analogs. There are two numbering systems for penicillins. According to USP, the nitrogen is numbered as position 1 and sulfur as position number 4. But according to chemical abstract system, sulfur is numbered as 1 and nitrogen is numbered as 4. When it comes to mode of action, penicillins first come in contact with the transpeptidase enzyme, then due to the highly strained beta-lactam ring, they are subjected to nucleophilic attack by the serine residue of the transpeptidase enzyme. So, this serine residue acts as a nucleophile, attacks on the carbonyl group of the beta-lactam ring and opens this beta-lactam ring, which leads to a formation of acyl enzyme intermediate. Here the penicillins irreversibly binds to this enzyme and forms a highly stable penicilloyl enzyme intermediate, and that is how it is going to inhibit the peptidoglycan biosynthesis. So, here the penicillin has a beta-lactam ring that is fused to 5-member sulfur containing ring called as thiazolidine. Without any acyl side chain here, this is called as 6-amino penicillinic acid. Now, this acyl side chain varies according to the components of the fermentation medium. The corn strip liquor was the first fermentation medium used for the biomass production of penicillins. It contains a component called as phenylacetic acid, and that is how you got penicillin G, which is also called as benzyl penicillin. Another fermentation medium contains phenoxyacetic acid, that is how you got penicillin V, which is also called as phenoxymethyl penicillin, and this is the first orally active penicillin. Here are some of the characteristic structural features of the penicillin. The strained beta-lactam ring is essential. The carboxylic acid at the second position is also essential. Usually, this carboxylic acid is present in ionized form, and that is the reason these penicillins are administered in the form of a sodium or potassium salt. Cis stereochemistry of these hydrogens is also essential. There is no substitution at 5th and 6th position. Sulfur is usually necessary but not essential. You can substitute this sulfur by a heteroatom such as oxygen or carbon. In all the cases, the penicillins would be as effective as the parent penicillin molecule. Bicyclic system is essential because it provides strain to the beta-lactam ring. The greater the strain, the greater is the activity of the penicillin molecule. But here is the catch: the greater the strain, the greater is the instability of molecule to several other factors, which we are going to discuss in detail in the next couple of minutes. Penicillin G is one of the first naturally isolated penicillin analog, and this is the structure of penicillin G, which is also called as benzyl penicillin, and the structure is just like a half open book. Penicillin G is used for variety of infections such as diphtheria, which is associated with severe nose and throat infections. It is also effective against sexually transmitted disease such as gonorrhea and syphilis. Penicillin G is also effective against meningitis, which is an infection caused by a bacterium called as Neisseria meningitidis. However, there are several drawbacks associated with this penicillin G. The first major problem associated with this penicillin G is it cannot be taken by a oral route because it is highly susceptible to the acids that are present in the stomach, which will lead to opening of this beta-lactam ring and making this inactive. It is effective only against the gram-positive organisms, so therefore it has a narrow spectrum of activity. And there are many infections against which this penicillin G is completely ineffective, especially those infections which are associated with bacteria that can produce an enzyme called as beta-lactamases. So, this beta-lactamases opens the beta-lactam ring of this penicillin G and making them inactive. Now, let us look how researchers like you and me came into picture with various strategies to tackle these problems and how these strategies led to the development of newer penicillin analogues with improved properties. So, as I mentioned you, one of the major problems associated with this penicillin G is its acid sensitivity. Now, there are three major reasons behind this acid sensitivity. The first reason is the highly reactive beta carbonyl group. The carbonyl group of this penicillin G is highly reactive and it is very susceptible to nucleophilic attack and does not behave like a normal tertiary amide. In case of normal tertiary amides, they are not electrophilic because here the carbonyl group is in resonance with the neighboring nitrogen. So, the nitrogen can donate its lone pair of electrons and thus forming this dipolar resonance structure with a bond angle of around 120 degree. This kind of resonance stabilization is impossible in a beta-lactam ring due to increased angle strain, which would result in having a double bond in a 4-member beta-lactam ring. The preferred bond angle for double bond is 120 degree, which is constrained to 90 degree in case of penicillin G. That is the reason why the lone pair of electrons is delocalized on a nitrogen atom only and the carbonyl group is more reactive than one would expect for a normal tertiary amide. Another major reason behind the acid sensitivity of the penicillin G is its ring strain. The beta-lactam ring is fused to this 5-member sulfur containing thiazolidine ring, and because of this bicyclic system it induces angle and torsional strain. Because of this reason, when you take this penicillin G via oral route, it can be easily attacked by acid that is present in the stomach, which can lead to opening of this beta-lactam ring. So, opening of the beta-lactam ring can relieve these angle and torsional strains. So, therefore, this is a more favored pathway for the degradation of the penicillin G. The degradation product that is obtained from this opening of the beta-lactam ring is called as penicilloic acid. Another major reason behind the acid sensitivity of the penicillin G is its participation of the neighboring group. So, here the acyl group actively participates in the opening of this beta-lactam ring, which leads to formation of inactive intermediates such as penylic acid and penicillinic acid. Therefore, penicillin G has a self-destruct mechanism built in its structure. It can be seen countering this problem of acid sensitivity is a very difficult task. Nothing can be done for the first two problems because beta-lactam ring is essential for its antibacterial activity. You cannot do any further modifications in a beta-lactam ring. However, the third problem can be tackled. This can be tackled by reducing this neighboring group participation, and how we can do that? By incorporating an electron withdrawing group. So, this electron withdrawing group will pull away the electrons from the carbonyl oxygen, reducing its tendency to act as a nucleophile, and that is how penicillin V was launched into the market. In case of penicillin V, it has a electronegative oxygen atom attached on the side chain of the beta-lactam ring, and therefore, it is more stable against acid when compared to penicillin G. Therefore, penicillin V can be taken via oral route because it is not susceptible to degradation by stomach acids. Another problem associated with this penicillin G is its sensitivity to the beta-lactamase enzyme. So, these are the enzymes which can open up the beta-lactam ring of the penicillin G, making them inactive, and this is one of the major defensive mechanisms that a bacterium brings to avoid these beta-lactam antibiotics. Again, these beta-lactamases enzymes can be classified based on the active site residue that is involved in the opening of this beta-lactam ring. Some contains serine residue at the active site that opens this beta-lactam ring, and in some cases they have a zinc metal which is involved in this opening of the beta-lactam ring. Whatever the type of beta-lactamase it is, ultimately it leads to the opening of this beta-lactam ring, and this is the product that is being formed, and this is called as penicilloic acid. However, this is not a stable product. It can undergo further decomposition. For example, it can undergo decarboxylation and it can give an intermediate called as phenyloic acid. This intermediate can undergo further degradation to give you phenylaldehyde and penicillamine. The problem of this beta lactamase became very critical in 1960s due to the widespread use of this penicillin G that led to the increasing rate of penicillin G resistant Staphylococcus aureus infections. However, the solution to this problem was just around the corner, and that is how the design of beta lactamase resistant penicillins came into picture. Here, the use of steric shield to block the access of penicillins to beta lactamase active site was a useful strategy, and this was achieved by incorporating a bulkier group. Again, here is the problem. If the group is too bulky, it can also restrict the access of these penicillins to the transpeptidase enzyme, which is its target, right. So, therefore, a great deal of work had to be done to identify an ideal shield which would allow the penicillins to access the transpeptidase enzyme, but block the access of penicillins to the beta lactamase enzyme. Fortunately, we got an ideal shield, and that is how methicillin came into the market. So, here methicillin has a methoxy group on the either sides, and that is how it is going to block the access to the beta lactamase enzyme. However, this is not an ideal drug because it does not have any electron withdrawing group here, and therefore, you cannot take this drug via oral route because it would be easily broken down by the acid that is present in your stomach. Therefore, this methicillin must be given via intravenous route of administration. However, this is not a clinical drug because some of the bacteria have developed resistance against this methicillin, and they are called as MRSA, which is abbreviated form of methicillin resistant Staphylococcus aureus. Therefore, it led to the development of other beta lactamases resistant penicillins such as nafcillin. Again, it has a naphthalene group here. This cannot be administered via oral route, it must be taken via parenteral route only. However, these beta lactamases resistant penicillins are kept as reserve troops and are introduced only into the fray when an infection develops resistant against many broad spectrum penicillins. Another major problem associated with this penicillin G is its narrow spectrum of activity. That means it is only effective against gram positive organisms, whereas against gram negative organisms it is completely ineffective. Now, the major reason for the spectrum of activity of for any antibiotic, here for example in case of penicillin, it depends on many factors, such as it depends on the ability of the penicillin G to bind with the transpeptidase enzyme, how efficient this penicillin G is to get inside the gram negative bacteria. Again, gram negative bacteria have defense mechanisms, they have efflux pumps which kicks out any antibiotic that gets inside a gram negative bacterial cell. Again, all these factors vary from bacteria to bacteria. Therefore, there is no clear cut tactic that can be used to increase the spectrum of activity of penicillin G. However, researchers found that the presence of hydrophobic group on the acyl side chain allows these penicillins to be effective against gram positive organisms. So, they thought, why do not we incorporate some hydrophilic functional groups like amino functional group and carboxylic functional groups, and they found that introduction of hydrophilic functional groups on the alpha position of the acyl side chain allows these penicillin molecules to be effective against gram negative organisms also, and that is how broad spectrum penicillins and extended spectrum penicillins came into the market. Here are broad spectrum penicillins such as amino penicillins and carboxy penicillins. In case of amino penicillins, you have an amino functional group on the alpha position of the acyl side chain. One such example is the ampicillin. Carbonicillin is an example of carboxy penicillin where there is a carboxylic functional group on the alpha position of the hydrophilic side chain. So, they are broad spectrum penicillins. That means they are effective against both gram positive as well as gram negative organisms. Another class of penicillins are called as ureopenicillins, and they are extended spectrum penicillins. Here in these cases they have a urea functional moiety that is present on the alpha position of the acyl side chain. They are effective against both gram positive as well as gram negative organisms, but also they are effective against some of the bacterial strains of a panel of escape pathogens. Here escape is the abbreviation of a panel of pathogens such as Enterococci, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter. So, here these iridopenicillins are effective against Pseudomonas aeruginosa, which belongs to this panel of escape pathogens. So, these escape pathogens are highly resistant to majority of antibiotics. Therefore, targeting these escape pathogens would be very cool thing. So, that is what these iridopenicillins do. Now, as you are familiar with many penicillin analogues, let us have a quick look at the classification of the penicillins. They can be classified based on a source it is obtained from. For example, penicillin G is a naturally obtained penicillin, whereas penicillin V is biosynthetically derived penicillin, whereas some penicillin analogues such as ampicillin and amoxicillin are man-made. Similarly, penicillins can also be classified into narrow spectrum penicillins, broad spectrum penicillins, and extended spectrum penicillins. The examples you have already seen previously, like ticarcillin and azocillin, these are extended spectrum penicillins which belongs to a family of iridopenicillins, whereas aminopenicillins and carboxypenicillins are broad spectrum penicillins, and methicillin, oxacillin, nafcillin, these are narrow spectrum penicillins. That means they are highly effective only against the gram-positive organisms. Again, these penicillins can be classified based on their acid susceptibility. For example, penicillin G cannot be taken via oral route because it is susceptible to stomach acids. Therefore, it is acid unstable, whereas penicillin V, it is acid stable, it can be given via oral route of administration. The same penicillin analogues can also be classified based on the route of administration. The penicillin analogues which can be given via oral route, they are acid stable, whereas the penicillin analogues which are acid labile, they must be given via parenteral route. For example, penicillin G is given by a parenteral route of administration, and that is the same thing that occurs even with methicillin and ticarcillin, etc. Whereas ampicillin and amoxicillin, they are acid stable and they can be given in the form of a tablets or capsule via oral route of administration. When it comes to side effects, penicillins are associated with common side effects such as nausea, diarrhea, dizziness, headache. But the most common side effect associated with this penicillin is allergy or hypersensitivity reactions, which can range from immediate response that can occur within minutes or hours to something that can happen days after taking the administration of penicillin. And these are called as delayed reactions, and this include skin rash or hives or anaphylaxis or bronchospasm or angioedema. So, bronchospasm is the process of constriction of airways of the lungs, whereas angioedema is associated with inflammation of face, mouth and lips. Anaphylaxis is a severe allergic reaction that can be fatal if not treated appropriately. Another major class of beta-lactam antibiotic is cephalosporins. Again, these are broad spectrum antibiotics and they are also bactericidal in nature. This cephalosporin C was the first cephalosporin that was isolated from Acremonium chrysogenium during the mid 1940s. In case of cephalosporins, they also have a beta-lactam ring which is fused to sulfur containing 6-member ring called as dihydrothiazine, and this is the basic code structure of this cephalosporin C or cephalosporin class of antibiotics, which is called as 7-amino-cephalosporinic acid. The side chain present in this cephalosporin C is the 7-amino-adipic acid. These cephalosporins are one of the most potent antibacterial agents. They are well tolerated and their development was paralleled that of the penicillins. If you look at the numbering system, according to chemical abstract system, sulfur is numbered as position 1 and nitrogen is numbered as position number 5. Now, like penicillins, these cephalosporins are also bactericidal and their target is also the same, that is transpeptidase enzyme. So, here also the cephalosporins comes in contact with the transpeptidase enzyme, and because of the strain in this beta-lactam ring, it is subjected to nucleophilic attack by the serine residue of this enzyme. So, here the serine residue acts as a nucleophile, attacks the beta-lactam ring and opens it. But here you have also a acetoxic group that is present in the third position of the cephalosporin family of antibiotics, and because of that it acts as a very good leaving group, and that is the reason it inhibits the transpeptidase enzyme irreversibly. And that is how these cephalosporins are going to inhibit the peptidoglycan biosynthesis. If you look at the structural activity relationship of cephalosporins, it has a beta-lactam ring which is fused to this 6-member sulfur containing dihydrothiazine ring. And now the strain present in this beta-lactam ring is less when compared to that of penicillins, but still this is effective to inhibit the transpeptidase enzyme. Again, the cis stereochemistry of hydrogen is essential. The sulfur is important, but it is not essential. For example, you can replace the sulfur by other heteroatoms such as oxygen, and in that case they are called as oxacephums. They are also as effective as the cephalosporins. Similarly, the sulfur can also be replaced by carbon, and in those cases those analogues are also effective like parent cephalosporins. But if the sulfur is undergoing oxidation to give sulfoxide or sulfone, the activity of these cephalosporins goes down. Similarly, the carboxylic acid at the second position is essential, and generally this carboxylic acid is in the ionized form, and therefore these cephalosporins are also administered as sodium or potassium salts. The double bond here is essential. If you remove this double bond, the cephalosporin loses their activity. Likewise, the acetoxy group is very important in terms of activity. It acts as a very good leaving group, and therefore these cephalosporins are going to inhibit the transpeptidase enzyme irreversibly. But again, the problem here is it is an ester bond, and because of that there are enzymes called as esterases present in your body which can open up this bond and making these cephalosporins less active. Therefore, this acetoxy group determines the pharmacokinetic parameters of the cephalosporins. Again, the acyl amino side chain is essential for the antibacterial activity of the cephalosporins. Like penicillins, if you can incorporate hydrophilic functional groups on the acyl side chain, they become more and more effective against gram negative organisms, and if there is a hydrophobic group, they will be effective against the gram positive organisms. But here there is one additional thing. There is an influence of the substance that is present on the acyl side chain. For example, iminomethoxy group, which is a substituent present in the cefuroxime, which is a second generation cephalosporin. Due to the presence of this iminomethoxy group at this acyl side chain, this cefuroxime is effective against bacteria that can produce beta lactamase. Because of this sterically hindered substituent, it cannot be cleaved by the beta lactamase enzyme, and therefore it is stable against many beta lactamases. Now, these cephalosporins can also be classified into different generations like first generation, second, third and fourth generation. Now, you may ask, is it relevant to know which cephalosporin belongs to which generation? And in short, the answer is yes, because each generation of cephalosporin tells you something about their spectrum of activity. For example, earlier generation cephalosporins like first generation, they are highly effective against gram positive bacteria, but they have diminished activity against gram negative organisms. But obviously they are broad spectrum antibiotics and their activity is less comparable to that of penicillins. But the mid generation cephalosporins like second generation and third generation, they are highly effective against both gram positive as well as gram negative organisms, and therefore they are broad spectrum cephalosporins. And the fourth generation is called as extended spectrum cephalosporins because they are effective against gram positive and gram negative organisms obviously. Along with that, it is also effective against some of the bacterial strains that are included in the escape panel of pathogens such as Pseudomonas aeruginosa and Klebsiella pneumonia, and therefore they are highly effective against many of the drug resistant bacteria. Now, again, each generation of cephalosporins can be further classified based on their route of administration. First generation cephalosporins like cephalothin and cefazolin, they have a basic structure like this. In case of first generation cephalosporins, they have a acetoxy group that is present at the third position. Because of that, it is metabolically very labile and it can be easily broken down by stomach acids. Therefore, this first generation cephalosporins like cephalothin cannot be given via oral route. It must be administered via intravenous route of administration. Now, the scientists thought, why don't we remove this acetoxy group? Instead of this acetoxy group, if we just keep methyl group here like this, then it is metabolically stable, and it turns out that it is well absorbed and therefore it can be given by a oral route such as cephalexin and cefaroxil. When we move from this first generation to second generation cephalosporins, they are highly effective against gram negative organisms, and they are also effective against some of the bacteria that can produce beta lactamases. The first generation cephalosporins are effective against gram positive bacteria, but less effective against gram negative bacteria, whereas second generation cephalosporins are highly effective against gram negative bacteria, but they have diminished activity against gram positive organisms. But a good thing is they are also effective against bacteria that can produce beta lactamase enzyme. And the same thing goes even with the third generation of cephalosporins. They are also effective against gram-negative bacteria, but with a diminished effect on gram-positive organisms. They are also effective against many of the bacteria that can produce beta-lactamases. And the additional advantage to this second generation and third generation cephalosporins are they are highly hydrophobic. Because of this reason, they can easily cross a blood-brain barrier, and that is why they are used for meningococcal infections such as meningitis. In case of fourth generation cephalosporins, they are effective against both gram-positive as well as gram-negative organisms. Along with that, they are also effective against Klebsiella pneumoniae and Pseudomonas aeruginosa. Because of that reason, they are called as extended spectrum cephalosporins, for example cefepime and cefepidome. So in a nutshell, as the generation of cephalosporin progresses, they become more and more active against the gram-negative organisms. Let us have a quick look at the structure of some of the generation of cephalosporins. For example, this is cephalothin, which is a first generation cephalosporin, and you can see there is a acetoxy group present here, which is very metabolically labile. Therefore, it cannot be given by a oral route. But when you go from first generation to second generation, it has a immunomethoxy group. Therefore, it is highly effective against gram-negative organisms and some of the bacteria that are producing beta-lactamases. Here, it has a urethane moiety. Because of that, it is metabolically stable. And the same thing goes even in case of third generation cephalosporins. But in case of third generation cephalosporin, the substituent present here is aminothiazole. Because of that, it is highly effective against gram-negative organisms as well as majority of the bacteria producing beta-lactamase enzyme. In case of fourth generation cephalosporins, there is a positively charged substituent present at the third position and carboxylic acid at the second position. Therefore, they behave as Zwitter ions at a physiological pH, and they are effective against Pseudomonas aeruginosa, Klebsiella pneumonia and many gram-positive as well as gram-negative organisms, and they are highly resistant to majority of the beta-lactamases. Like cephalosporins, they are also used for variety of infections such as urinary tract infections, respiratory tract infections. They are also effective against typhoid and bacterium called as Neisseria meningitis, which is a causative agent for meningitis. They have a common side effect, that is allergy or hypersensitivity reactions. Let us move to another class of beta-lactam antibiotics, which are monobactams. The name itself suggests these are unique beta-lactam rings which are monocyclic and they are not fused with any other ring. Again, these are narrow spectrum antibiotics and they are bactericidal in nature. So here the monobactams are highly effective against gram-negative organisms as well as Pseudomonas aeruginosa. They are preferred for infections such as hospital-acquired pneumonia, meningitis, urinary tract infections and osteomyelitis, which is associated with the inflammation of bone of arms and legs. Like penicillins, they have a common side effect of skin rash and abnormal liver functions. And the class of these beta-lactam antibiotics or carbapenems, they are broad spectrum antibiotics and they have bactericidal nature. They have a 4-membered beta-lactam ring which is fused with a 5-member carbon containing ring, and that is why they are called as carbapenems. So some of the well prescribed carbapenems include imipenem, meropenem, etapenem, dorapenem, etc. Again, like monobactams, they are highly effective against gram-negative organisms including Enterobacteriaceae. They are preferred for infections such as hospital-acquired pneumonia, meningitis and intra-abdominal infections. The carbapenems are associated again with a common side effect of allergy, but one of the another side effect that is observed with the use of imipenem and meropenem are seizures, which are also called as epilepsy, which is characterized by symptoms of abnormal muscle spasms, abnormal bladder and bowel movements and blackout followed by a period of confusion where the patient cannot remember for a short period of time. One of the major problems associated with many beta-lactam antibiotics is that they are highly susceptible to the enzyme called as beta-lactamases, which is produced by a bacteria in a defensive way to get rid of these beta-lactam antibiotics. So now scientists thought, why don't we make inhibitors of this beta-lactamase enzyme itself, and that's how these beta-lactamase inhibitors such as clavulanic acid and sulbactam came into the market. So here these inhibitors irreversibly bind to the beta-lactamase enzyme and inhibit them, and therefore when these beta-lactamase inhibitors are given in combination with beta-lactam antibiotics such as penicillins or cephalosporins, they will show synergistic effect. Here are such examples. Amoxicillin is always given in combination with the clavulanic acid, which is a beta-lactamase inhibitor, and the same thing goes along with sulbactam. For your information, these are some of the bacteria that can produce beta-lactamase, and therefore these bacteria are very difficult to target by many beta-lactam antibiotics: methicillin-resistant Staphylococcus aureus, Klebsiella pneumonia, Enterobacteriaceae, and Staphylococci. Let's take an example of beta-lactamase inhibitor, clavulanic acid. This was first isolated from Streptomyces clavigelis, and it is an irreversible inhibitor of beta-lactamase. Clavulanic acid is given in combination with amoxicillin, and this is the structure of clavulanic acid, where the 4-member beta-lactam ring is fused to oxygen containing 5-member saturated system. The strained beta-lactam ring is essential. The carboxylic acid at the second position is also essential, because again this is present in the ionized form, and clavulanic acid is also given in the form of a potassium salt. If you notice here, there is no substitution at the 6th position. The stereochemistry of the hydrogen and carboxylic acid must be in R configuration. So, there is a enol ether present at the 3rd position. The configuration is Z for a double bond, and the hydroxyl group present at the 9th position is involved in the hydrogen bonding interaction with the residues of the beta-lactamase enzyme. First, the clavulanic acid comes in contact with the beta-lactamase enzyme. Due to the strained beta-lactam ring, it is subjected to nucleophilic attack by the nucleophile of this beta-lactamase enzyme, which leads to opening of the beta-lactam ring. This is further followed by the attack of nucleophile of the enzyme, and thus it binds to drug irreversibly. And this is how clavulanic acid irreversibly inhibits the beta-lactamase enzyme. Let us move to another class of antibiotic which inhibits the cell wall biosynthesis, but by a different mode of action. For example, cycloserine. Cycloserine is a broad spectrum antibiotic, and it was first produced by a fungus called Streptomyces gariphelus. It has a unique mode of action. If you remember, I told you the peptidoglycan biosynthesis is mediated in 3 phases, and the initial phase is called as a cytoplasmic phase, where the D-alanine unit is being formed. This is formed from an enzyme called as alanine racemase, which converts the alanine to D-alanine. Further, these D-alanine units are ligated in the presence of D-alanine ligase, and once this D-alanine unit is being formed, it is then added to the peptidoglycan component, and that is how the peptidoglycan is being synthesized. Cycloserine targets this alanine racemase and D-alanine ligase, and that is how it is going to inhibit the cytoplasmic phase of the peptidoglycan biosynthesis. The D-cycloserine is prescribed for urinary tract infections, and it is the second line agent in the treatment of tuberculosis. Obviously, it is given in combination with other anti-TB agents to target the Mycobacterium tuberculosis. Another class of cell wall biosynthetic inhibitor includes glycopeptide antibiotics. As the name itself suggests, they contain tricyclic or tetracyclic glycosylated heptapeptide codes. This is the macromolecular structure, and again they can be classified based on the R and R1 substituents. Type 1 glycopeptide antibiotics have asparagine or glutamine at the R1 and valine at the R position. Type 2 antibiotics have aromatic residues on the R and R prime, but they are not connected with each other. But in case of type 3 glycopeptide antibiotics, the 2 aromatic residues are connected via ether bridge. In case of type 4 glycopeptide antibiotics, they have 2 aromatic residues attached with a ether bridge. Along with that, they also contains fatty acid as well as amino sugar. One such example of glycopeptide antibiotic is the vancomycin. It is a narrow spectrum antibiotic, but it has a bactericidal effect. That means it is going to kill the bacteria. So it has a unique mode of action. Like penicillins, vancomycin is also going to inhibit a transpeptidase enzyme, but along with that, it also inhibits the transglycosylase enzyme. Because of this macromolecular structure, vancomycin caps the pentapeptide tail by hydrogen bonding, and thus it acts as a steric shield, blocking the access of this pentapeptide to membrane bound enzymes such as transglycosylase and transpeptidase. That is how it is going to inhibit the peptidoglycan biosynthesis in the extracytoplasmic phase. Vancomycin is mostly prescribed to treat methicillin resistant Staphylococcus aureus infection. Unfortunately, few of the bacterial strains also have developed resistance against vancomycin, and they are called as VRSA, vancomycin resistant Staphylococcus aureus. To summarize, we have talked about various antibiotics that inhibit the cell wall biosynthesis. So either they are going to inhibit the cytoplasmic phase, or they are going to inhibit the extracytoplasmic phase of the peptidoglycan biosynthesis. For example, D-cycloserine is going to inhibit the alanine racemase and D-alanine ligase. That is how it inhibits the cytoplasmic phase of the peptidoglycan biosynthesis. But once the peptidoglycan components are biosynthesized, they are translocated to the extracytoplasmic phase, where they are assembled in the presence of transglycosylase and transpeptidase enzyme. Transglycosylase enzyme is the target for vancomycin, and transpeptidase enzyme is the target for both beta-lactam antibiotics as well as vancomycin. That is how all these antibiotics inhibit the cell wall biosynthesis. Let us move on to another class of antibiotics that target the cell membrane, and these are called as polypeptide antibiotics. As the name itself suggests, they have a polypeptide structure. These are bactericidal in nature. They are highly effective against gram-negative organisms. Some of the commonly prescribed polypeptide antibiotics include agents such as bacitracin, polymyxin, and colistin. Here are the sources of various polypeptide antibiotics. Bacillus polymyxa is a source for the polymyxin B, bacitracin is obtained from Bacillus subtilis, and colistin is obtained from Bacillus colistinus. Generally, these polypeptide antibiotics are given in the form of topical agents because they cause systemic toxicity. For example, bacitracin is given for local infections in the form of topical agents, and colistin is used for urinary tract infections. This is a structure of polymyxin B, and if you see, it has a 2,4-diaminobutyric acid residues, because of which it exists as a polycation at a physiological pH. Polypeptide antibiotics have a unique mode of action. They have detergent like action on the cell membrane, and they have high affinity to the phospholipids and peptide molecules, which results in a membrane distortion and pseudopore formation. This leads to a leakage of cellular constituents followed by cell lysis and death of the bacteria. So therefore, these polypeptide antibiotics are bactericidal because they are directly killing the bacteria by disrupting this cytoplasmic membrane. So in a nutshell, in this lecture, we have talked about the antibiotics that target the cell wall biosynthesis, which include beta-lactam antibiotics such as penicillins, cephalosporins, cycloserine, and glycopeptide antibiotics such as vancomycin. We also talked about another class of antibiotics that disrupt the cell membrane and kills the bacteria, and those are called as polypeptide antibiotics. So in the next upcoming lecture, we will talk about another class of antibiotics which either inhibit the nucleic acid biosynthesis or protein biosynthesis or metabolic pathways. Thank you.